Welcome along, everyone. Good afternoon, and welcome to the latest in our series of webinars. My name is Moi University. I'm a lab engineer here at BioReperia, and today I will be moderating this webinar. Today's focus will be on breast cancer, and today we have two speakers. The whole webinar will go on for approximately 45 minutes, at the end of which we will have a Q&A session. I would invite you all to put your questions you might have in the chat and we'll get to them. Alternatively, if you think of some questions you want to ask us at a later date, then please send us an email and we'll love to answer them for you. This webinar will be recorded and we will upload it onto our YouTube channel, so you can watch it later. You can share it with friends and colleagues. But we will also ask that you keep your camera and your microphone turned off. And that's all the rules we have. And so I will now introduce our two speakers. I hope you forgive me. I'll be using notes because I'm a stickler for the details. So first up, we have Gabriela. She is a project manager at BioReperia. She has a master's and a PhD in molecular biology and biotechnology, and she did her postdoc here in Linköping in Sweden, where she studied the molecular mechanisms in the tumor microenvironment that drive breast cancer progression, angiogenesis, and metastasis. Her research used different techniques, which included spheroid cultures and the Zebrafish tumor xenograft model, where she accumulated more than 10 years of experience working with the Zebrafish model with cancer. Our second speaker today is Dr. Rispah Torrorey-Sawe. She is a senior lecturer in immunogenetics and tumor immunology at Moi University's Department of Pathology and Immunology section. She holds a bachelor's in science, a master's in immunology from Moi University, and a PhD from Maseno University, where her research focused on aggressive breast cancer in Western Kenya. With postdoctoral training in Stellenbosch University, she specializes in personalized genomic medicine, implementing multi-gene assays and establishing ethical frameworks for genomic research. Dr. Torrorey-Sawe's work addresses critical disparities in breast cancer, particularly among Black women who face higher rates in aggressive subtypes and significant barriers to care. Currently, she collaborates with researchers in Sweden, America, and Africa, and where she tackles these issues. In recognition of Breast Cancer Awareness Month, she is committed to improving survival rates for Black women with triple-negative breast cancer through innovative therapies and Zebrafish models, position herself as a leading advocate in the fight against breast cancer disparities. So, with no further ado, I will lead you over to our first speaker, which will be Dr. Rispah.
Thank you very much, Mike, for your kind introduction. I'm going straight to our webinar today. The title is Zebrafish Models for Breast Cancer Research and Immunology Focus. So this is what we expect to go through by the end of this webinar. We'll look at a bit of breast cancer epidemiology, breast cancer immunology, the challenges in breast cancer immunotherapy, and then my co-presenter will look at the preclinical models overview, how do we address the challenge, and also spend some time to make you people understand zebrafish in immuno-oncology and perspectives. So to start with, let's look at the breast cancer globally, the incidences. So breast cancer is the second most common cancer worldwide. And according to WHO, close to 2.3 new cases were reported in 2022. The other thing is that breast cancer is common in many countries. Actually, it's the leading cancer in 157 countries out of 185, as you can see in the map. Most of these are breast cancer. The pink indicate that breast cancer is the leading cancer. And then breast cancer also causes mortalities close to 700 100,000 globally. And this is still 2022 WHO data. And roughly half of the breast cancer occur in women with no specific risk factors than sex and age. Again, there is approximately 0.1 to 1% of male breast cancer, which also should be included in our perspectives. So the other thing is to look at what my interest is, and that is breast cancer disparities. We seem to have high it is recorded a high incidence of breast cancer in high-income countries, that is in North America, Western Europe, than in low and middle-income countries. I mean, that is in Africa, sub-Saharan Africa. And in the last 40 years, breast cancer outcomes have improved in the high-income countries. And if you look at the 5-year survival rate, it goes up to at least 85% compared to 60%, and sometimes it's even lower for the low income countries. So that is the disparity that we talk about. And this is many factors contribute, including the genetic predisposition, tumor characteristics across populations. Next is to look at the tumor microenvironment. So tumor microenvironment consists of a complex that involves the stroma part, immune part, and also the extracellular matrix, as you can see in this diagram. So the reciprocal and dynamic interaction between the cells and between cells and the extracellular matrix contribute to the tumor evolution and also the heterogeneity that is seen in breast cancer. And conversely, the tumor remodels itself in the environment. And this is to avoid the immune responses, which is really sometimes strong, but it has evolved ways of eliminating the immune response. So tumor microenvironment can possess tumor antagonizing or tumor promoting functions, and these impact breast cancer therapies. So to understand tumor microenvironment, we do this by looking at what is innate, especially the tumor, the infiltration of the immune cells. So in the tumor microenvironment, it is infiltrated by a diverse array of immune cells. And when I talk of immune cells, I mean all the immune cells from T, B, natural killer cells, dendritic cells, macrophages, and even myeloid-derived cells. So the balance between the effector immune cells, meaning the immune cells that can directly kill the tumor, or the ones that can cause immunosuppressive function, like, for example, the inhibiting anti-tumor immune responses. All this, understanding this within the tumor microenvironment is critical in determining tumor progression and also response to therapy. So when you look at breast cancer or any other cancer immunity, you start from innate to adaptive. So all the time, our immunity will recognize the non-self part, meaning the part that was not seen during development. And that is when, if anything is seen that is not really self, then the first part of the immunity that plays or kicks in is the innate immunity. And then innate immunity will always communicate to the adaptive immunity. And when you look at the innate immunity, the key players include antigen-presenting cells, the natural killer cells, macrophages, and it will call for help from the adaptive immunity, and that will involve antigen presentation, and then the T cells will see. When the T cells have been activated, then it can kill the tumor directly or use the other pathways that involve differentiation into different subpopulations, releasing cytokines that will give the effector function. But tumors have really evolved ways of evading immunity by releasing cytokines that create immunosuppressive microenvironment. For example, inhibiting of CD8 positive T cells and natural killer cells, and also promoting of regulatory T cells and tumor-associated macrophages. One of the cells that will be very important in this webinar. They also then regulate antigen presentation as a way of evading the immune function. And you know, the immunity can never, some part of the immunity, especially the adaptive immunity, the T cells cannot see antigens unless they are presented via MEC molecules. So it's really important to have all these things functioning well for a potent immune response. So the other thing that maybe we need to really bring to our attention is the tumor-associated macrophages that seem to exist in a plasticity situation where you either have a pro-inflammatory group of macrophages or classical activated macrophages and the alternative activated macrophages. So the M1 macrophages are the most potent, just like the Th1 immune response, while M2 macrophages is alternatively activated macrophages that is seen in immunosuppressive tumor microenvironment. So we've done several studies in our Western Kenya population. We've looked at the role of immune responses in the aggressive forms of breast cancer. We were able to look at tumors of breast cancer patients and then compare with a non-tumor. And we could see increased infiltration of the M2 macrophages, meaning the CD163 positive. We also recorded the CD25 positive regulatory T cells and not the B cells, and also increased CD8. So there is a lot of questions because in Africa, we tend to see aggressive forms of cancers and the immune system could be playing a role. So there is a lot that needs to be done to really understand. So in summary, what we came up with in our conclusion is that our breast cancer tissues exhibit high expression of T cells and macrophage markers, especially the M2 macrophages and Tregs, compared to the median tissue, and there is need to really look into this. So this diagram is summarizing what happens with the immune response. It starts from a normal cell, and then carcinogenesis takes place in the cell cycle. And then you have a state where the immune system can still eliminate all the possible tumor antigens. And if that is not possible, it moves to the second phase, that is equilibrium phase. In the equilibrium phase, you have tumors really changing their surface markers and trying to evade the immune system. But if proper management is taken or even lifestyle is changed, this can go back to the elimination phase. And if that mutations, for example, accumulate over time and tumors succeed in evading the immune response, then it goes to the last phase called the escape phase, where there is metastasis and so on. So this has really informed the targets that have been used for immune-based therapies, as we can see in the next slide. So here we have a diagram that is showing all the possible immune-based therapies that have been tried or FDA approved in breast cancer. And from the breast tumor, you always have the immune response that is starting with antigen presentation via the MSC molecules, and then T cells will be activated, and the cells that play a role is mature dendritic cells, which play a role both in adaptive and innate immunity, and then natural killer cells and macrophages in an innate manner, then antigen presentation, where adaptive immunity will be activated, and you have your effector B and T cells. And all these, they are targets that have been done for immune-based therapies. Still, this is more on immune-based therapies. We have checkpoint inhibitors. Examples are CTL4, anti-CTL4, and anti-PD-1. We also have Group B, that is CAR T-cell therapies. We have Group C that will work against the viruses. We have vaccination that is coming up, and we have T-cell receptor T-cell therapy. So challenges in breast cancer immunotherapy there, although it's really promising, but there's still challenges. The first one is the variability of patient response to the immune checkpoint inhibitors, and also the difficulty in identifying reliable predictive biomarkers, and also understanding the immune landscape within these tumors. So I'll stop here, and Michael's presenter will continue.
Thank you, Rispah, for a very nice background, very informative. And yes, so to address this challenge, we really need in vivo preclinical models that can recapitulate the tumor microenvironment faithfully, and that's the most important. And right now, the mouse tumor model is the model that is most commonly used in research because of its many benefits. But even though the mouse model, most of mouse model, tumor model, they don't include the human microenvironment. And to address this challenge, humanized models have been developed. However, this type of mice, they are costly, time-consuming, and they have ethical concerns. And as well, evaluation of metastasis, which is most important, is very difficult to evaluate in the mice model. That's when the zebrafish comes into place, especially in the embryonic stage. The zebrafish model is quite fast. It's only 5 days as a duration. Because it's very small, it's cost-effective as well and scalable, and has a high throughput screening. And since it's transparent, one can evaluate the interactions between the tumor cells and the immune cells, how are their interactions in the microenvironment, and it's possible to have a complex microenvironment as well. Metastasis, the metastatic process is faithfully recapitulated in the zebrafish model, and that is why it makes a very good complement to the mouse model as an in vivo preclinical assays. And it has an ethical benefit because at the embryo stage, the zebrafish is not considered an animal, so there are no need for an ethical permit to work with the zebrafish model. So to deep dive a little bit more in the zebrafish tumor xenograft model, I will start by explaining how this model works. So everything starts with cell suspension of tumor cells. These tumor cells are labeled with a red fluorescent dye, and they are implanted into these old zebrafish embryos, as you can see here in the image of the tumor cells being implanted. After implantation, the zebrafish larvae are placed in multiple plates, one larvae per well, to evaluate each embryo separately through the evaluation time. The treatment, for example, chemotherapy, can easily add it to water, and the zebrafish embryo will absorb that treatment through the skin and the gills and through the gastrointestinal tract. Other type of treatment, like immune therapy treatment, like antibodies or cell therapy treatment, can easily combine together with the tumor cells and co-implanted together, or can be administered intravenously. So after 3 days of treatment, there are 2 outcomes that can be evaluated in the zebrafish model. The first one is the primary tumor evaluation. And in here you can see in the upper panel how a primary tumor is observed in the control zebrafish embryo, that after 3 days the primary tumor size is roughly the same, while an embryo treated with a highly efficacious compound, after 3 days is just barely some cells left compared to the primary tumor at the day of implantation. Another output, and the most interesting one as well, is evaluation of metastasis. This metastasis can be evaluated in the zebrafish tail in a region that is called the caudal hematopoietic plexus. In here, metastatic cells established metastases, and we can count them manually. And the number of cells that we observe here in the tail faithfully recapitulate the metastatic potential of the cells that are implanted in the zebrafish embryo. In the next slides, I'm going to show you 3 different studies where the zebrafish tumor xenograph model has been used to elucidate or trying to elucidate the immune mechanisms that drive breast cancer metastasis. And for the study, to start studying the type of immune cells that drive this response, I will start by talking a little bit more about macrophages. In this slide, we can see data, human data, in the first graphs. This data was gathered by using this technique that is called microdialysis. So one catheter, it's implanted in the breast cancer tissue, and another catheter in the adjacent normal breast. So by doing this technique, we could detect 2 cytokines, CCL2 and CCL5, that were highly expressed in the tumor tissue, but not in the normal adjacent breast. Pathology or immunohistochemistry data showed that there is a huge infiltration of macrophages in the breast cancer tissue compared to normal breast. Mice models, the PYMT mice model, showed that in response to estradiol, tumor growth it's induced, not surprisingly. And the mice model helped us to observe that estradiol increases significantly the expression of this or the secretion of these cytokines, CCL2 and CCL5. We can see that by immunohistochemistry also in the mice tumors that the infiltration of macrophages into the primary tumor is quite high. And we could corroborate that this is because of the estradiol signaling that these macrophages were infiltrating the primary tumors, because when adding treatment with tamoxifen, this infiltration was significantly reduced. We also evaluated that these infiltrated macrophages were of the M2 class or protumoral macrophages, because analysis showed that these macrophages had a high expression of the MRC1 marker, which is a characteristic of M2 macrophages, and also the low expression of the INOS as well, a very characteristic of M2 macrophages. To evaluate whether these macrophages played a role in tumor metastasis of cancer cells, we went to our zebrafish model, and we implanted MCF7 cells, which are a model of the luminal A breast cancer. And we observed that estradiol treatment by itself didn't increase the dissemination of cancer cells. However, co-implantation of human monocytes with tumor cells significantly increased the dissemination of cancer cells. And this increase was further enhanced in presence of estradiol and human monocytes. In order to evaluate whether this dissemination was because of this CCL2 and CCL5 signal of these cytokines, using the zebrafish model, we added some antibodies blocking the effect of CCL2 and CCL5, or the mixture of both. And we could observe that this treatment, antibody treatment, could revert the metastatic ability of the cancer cells. So we can see that by this study, we can conclude that it's the CCL2 and CCL5 cytokines are the ones that mediate the macrophage-dependent dissemination of breast cancer cells in response to estradiol, bringing to CCL2 and CCL5 as promising targets for immune therapy. Moving forward to a different immune cell, such as the neutrophils, especially because since the neutrophils are the first responders in an inflammatory response condition like in cancer, by doing this, again, the microdialysis, we observe that in the breast cancer tissue of human in patients, there is an increase in the secretion of TGF-beta1 cytokine, which is an important cytokine also for the neutrophils and for the immune response in general, compared to the normal adjacent breast. We evaluated 2 different mouse models. The PYMT mouse model showed that in presence of estradiol treatment, there was we could notice more neutrophils here in the primary tumors, and this presence of neutrophils was significantly reduced in presence of fulvestrant treatment. Another mice model corroborated this. This mice model was for MCF7 xenografts. And we can see that here with the estradiol, there is a massive amount of neutrophils, which was also reduced in presence of fulvestrant. So we can say that also the estradiol played a very important role in recruiting the neutrophils. And what's most interesting is that these neutrophils were placed in the invasive front of the primary tumors. On the contrary to the macrophages, as we observed previously, that the macrophages infiltrate the primary tumor, the neutrophils stay in the invasive front. To evaluate whether the neutrophils played a role as well in the metastasis of breast cancer cells, we used our Zebrafish model, where we implanted MCF7 cells, and we observed the same response as before, that estradiol didn't really affect the dissemination of breast cancer cells. However, co-injection of human neutrophils together with tumor cells increased significantly the dissemination, and again, this was further increased in presence of estradiol. So it was very, very similar, or as we observed with the macrophages. By looking into the transparent embryo and by labeling the neutrophils in blue and the cancer cells in red, we could observe that in the metastatic niche, there are tumor cells and neutrophils co-migrating here together and interacting in the metastatic area. And by looking at the primary tumor, we could observe the same, in which both tumor cells and neutrophils could co-migrate together in the primary tumor. So giving an indication that these cells are closely interacting close to each other. And possibly this could be a response of integrins. And one of the integrins that it's specific for neutrophils is LFA1 integrin. We perform immune fluorescence in the mouse tumors, and we could see that those neutrophils that are precisely in the invasive front of the primary tumors, they had a high expression of this LFA1 integrin, which this expression was reduced in presence of fulvestrant. To further investigate these mechanisms, we isolated human neutrophils and we performed cultures of these neutrophils and treat the neutrophils with different treatments. We observed that the treatment of the neutrophils with estradiol or recombinant TGF-beta increased the LFA1 integrin in the neutrophils. And treatment with fulvestrant restored the expression of LFA1 integrin in the neutrophils. So we went back to our zebrafish model to evaluate whether this LFA1 and TGF-beta played a role in mediating the neutrophil-dependent dissemination of breast cancer cells. And indeed, by using antibodies against LFA1 and TGF-beta, it was the dissemination, the neutrophil-dependent dissemination of breast cancer cells was reduced significantly. And this, we corroborated this same data in a different zebrafish breast cancer model with a different cell line, which both treatment was successful. I mean, the dissemination was again significantly reduced. So by in this study, we could observe that TGF-beta was mediating the cell interactions and the neutrophil-dependent dissemination of breast cancer cells in response to estradiol in the tumor microenvironment. So the zebrafish can also be used to evaluate the mechanism of treatment resistance in breast cancer. In this slide, I would like to show you some data with PDX models that we performed this study in collaboration with Charles River. Since these PDX models are highly characterized, so it can help us to know what the mechanisms underlying this the resistance of treatment resistant. So we received mice tumor tissue from Charles River that it was clinical samples that were previously expanded in the mice, and this tissue we dissociated and labeled and implanted in the zebrafish larvae, which we treated with standard of care, in this case docetaxel and trastuzumab. We evaluated 20 different PDX models, and 19 out of 20 could be successfully implanted in the zebrafish model, having a take rate of 95%. And by using the zebrafish model, we could detect the responders here in green and the non-responders here in red. And we observed that the drug response was correctly predicted for 9 out of 11 PDX models with a sensitivity of 82%, and 8 of 11 predicted responders were also responded in the mice, showing a specificity of 73%. And not only that, we also observed that there was a very good correlation between the tumor grade in the patients and in the number of metastasized cells in the zebrafish PDX models, as you can see here in the graph. And this same correlation has been published by other research groups, where zebrafish avatars really can see as a good prognosis tool, since stage 4 patients also highly disseminate in the zebrafish compared to those patients in earlier stages. So by taking this data and all the information from these PDX models, such as RNA sequencing and microarray data, we could see that the models that were resistant to treatment, to trastuzumab and docetaxel treatment, had a higher expression of these 3 proteins, having this IL-2 or interleukin-2 receptor alpha being the most highly upregulated in trastuzumab, in docetaxel and trastuzumab resistant PDX model, when combining both RNA-seq and microarray data. So having this information, we evaluated an immune therapy, an antibody that is called daclizumab, that targets interleukin-2 receptor alpha. So we evaluated again one of the resistant PDX models in the zebrafish. We performed the dissociation, and for the single cell suspension, we combined with the daclizumab treatment, and we co-implanted in the zebrafish embryos. So after 3 days of treatment, we evaluated the primary tumor size, and we observed that the treatment with daclizumab restored the sensitivity to docetaxel and trastuzumab. So until now, I have been talking about how we can use the zebrafish model to evaluate the innate immune system in breast cancer metastasis. However, the zebrafish can also be used to evaluate the adaptive immune system, like evaluating cell therapies or tumor-infiltrating lymphocytes, and also to evaluate how other cancer-associated cells can participate in this process, like adipocytes or cancer-associated fibroblasts. Immune checkpoint inhibitors, as well as other immune therapies and treatment combination with the standard of care, can also be evaluated. As well, since the zebrafish model has shown a very good prediction of treatment outcome, so it can be used as a tool for precision medicine in the clinic. And since the metastasis that is observed in the zebrafish correlates to what is observed in the clinic, so the zebrafish can also be used as a good prognostic tool for the metastatic disease in patients. And having this information, and as Rispah was mentioning in her presentation, so we are collaborating with each other to evaluate clinical samples from the Moi Teaching and Referral Hospital in Kenya, where we are going to perform a research and development study testing chemotherapy alone and in combination with immune therapies in breast cancer clinical samples, first from a triple negative breast cancer, which the need is primarily there, but then it will extend to other breast cancer subtypes and also to other African countries. Once doing this, the next step will be a clinical implementation of the zebrafish tumor xenograft model as a precision medicine tool to evaluate the best treatment for each patient, and also whether each patient will benefit for whether only the chemotherapy or a combination of chemotherapy and immune therapy, with the ultimate goal of bringing this zebrafish technology close to the patients. Yes, so thank you very much for your attention. This, as Mike mentioned, this is a part of a webinar series. These are our past webinars that if you weren't able to look at them or to attend them, they are available in our YouTube channel. I invite you to check it out and take a look at these past webinars. This webinar will be also available in YouTube. And I invite you to our upcoming webinar. This will be announced as well in our LinkedIn webpage. Our next webinar will be in November the 7th, which will be co-hosted together with In Vivo Biosystem. And the last webinar of this webinar series will be by the end of November, and this will be mostly about prostate cancer, and this will be in collaboration with an experienced researcher from Mount Sinai. So yes, thank you very much.
Excellent. Oh, thank you very much, both of you. Extremely interesting talks. And we now will open it up for some questions from those watching. So if you have any questions or anything you'd like to know, then please put them in the chat and I will pass them on. But I'm going to ask a question myself, if that's okay. So Dr. Rispah, you focus a lot on aggressive breast cancer. I wonder if you could briefly define what aggressive breast cancer is, and perhaps what are the causes that cause the aggressive subtype?
Yeah, thank you very much, Mike. There are many ways in which you can classify breast cancer into aggressive, but the easiest is to do characterization using the 3 markers. That is ER marker, that is estrogen receptor, progesterone receptor, and HER2 receptor. So if you can use the 3 receptors to classify breast cancer into either lumino A, B, or triple negative, which is more of basal-like. So triple negative in African population seem to be higher than any other population, all factors taken into consideration. And it is one of the trickiest cancers to treat because there are no targeted receptors like in ER and HER2 receptors. And therefore, because of lack of receptors, patients mostly are put on chemotherapy, and prognosis most of the time is not good. Yeah. So, and given the fact that most of our patients come late to the hospital when the cancer has metastasized, for example, and we never test which chemotherapy are sensitive for the patient before treatment. So I think bringing a Zebra model close to the patients, especially to the African population patients, will really help our clinicians to make a decision on which treatment to go for, and that eventually may actually improve prognosis.
Absolutely.
Yeah.
Thank you. That's very interesting. One question here. Is it possible to evaluate the role of other signals in the tumor microenvironment that promote cancer metastasis other than estradiol?
Yes, of course. There is we can evaluate other signals, such as, for example, nutrients. There has actually been a different study that we, because of the time, we couldn't present, but we have also evaluated amino acids, for example. We have seen that amino acids also play a very important role in the metastasis of breast cancer. We can play with a lot of treatments with the cancer cells. We can evaluate the different knocking down some genes in the tumor cells and see how they respond to all of these other signals in the tumor microenvironment and how this affects their metastatic dissemination. But here in this particular study, we observed that lysine, for example, this amino acid played a very important role in dissemination. So, yeah, it's the possibilities are endless. I mean, it's there are many opportunities to evaluate.
That sounds great. Also, I had some thoughts while listening to you present regarding the immune system and its role in cancer. It seems that sometimes it can help, but then sometimes it can also harm. Is this the case?
Yes.
You've seen.
Yeah, the immune system actually is like a double-edged sword. Yeah, it can protect you, and at the same time, the same immune system can be activated to harm. Like, for example, tumors have really evolved ways of making use of the immune system for their own good. And one of the ways is actually taking advantage of the inflammation process so that it can, for example, cause angiogenesis for supply of oxygen and also nutrients. So it's true, immune system can protect us against tumors, but at the same time, tumors can also use the immune system to protect themselves. And in that case, it will harm the patient or the host. Yeah.
That's really interesting. A lot to think about. We have a question just coming in. Have you used molecules such as PD-1 or immunocheckpoints in a zebrafish model, and what was the outcome? Do you have a similar response as in murine models?
Yes, we have used this, and yes, what we have observed is that when we use patient material and implanted in the zebrafish, and at the same time we use this tumor-infiltrating lymphocytes, and we administer together with this anti-PD-1 treatment, we can see that the tumor-infiltrating lymphocytes by itself, they have a cytotoxic activity. But in presence of this anti-PD-1, this cytotoxic activity is further enhanced as well. So, and since in the because of the transparency of the zebrafish model and the amenability of the zebrafish model to implant immune cells intravenously, we could observe that those immune cells, those tumor-infiltrating lymphocytes, find the primary tumor and started to infiltrate the primary tumor in the fish. And this infiltration was even further increased in presence of the immune checkpoint inhibitor.
Okay.
So in this way, we can see that it's also very similar to what is observed in the mice models. Yeah.
Oh, fantastic. So yeah, very comparable then. I have a question here. So you mentioned that the zebrafish will be used to predict patients' response. Has this been validated, and how transferable is the zebrafish model to patients? And how does the zebrafish model compare to organoids? So we talked about how does it compare to mouse now.
Yeah, how it compares to human. Yeah, of course, yeah, we have validated this model with different types of indications, with bladder cancer, ovarian, colorectal as well. And we have seen a very good prediction of treatment outcome, around 80% prediction of treatment outcome to what it's observed in the clinic. So that's why we're Rispah it's collaborating as well, because that's what we are looking for to establish in Kenya and other African countries. And yes, and there was about the organoids, right? Yeah, and the organoids. Well, yeah, I mean the organoids and the zebrafish can also be very complementary models, because the organoids have also showed prediction, a higher prediction of treatment outcome as well in patients. But one limitation, or some of the limitations of the organoids, is that it's not possible to have the vasculature in the organoids, or vascularization. So all those treatments that kind of participate in the homing process cannot be evaluated in the organoids, and of course the metastasis, the metastatic process.
Yeah.
But yeah, as I mentioned, together with the mice models as well, all these preclinical models are complementary to each other. And so the best way moving forward is just take advantage of all these benefits that each model represents.
Yeah.
So that
it can all exist in the pipeline. For sure. Okay. I think we're going to have to draw the session to a close. We've run on a little bit. If I can just click on to the next slide. So I would urge you to contact us if you have any questions. We always like to answer your questions and hear from you. But last thing is just to say thank you very much for Dr. Rispah. It's been wonderful having you visit with us, and you're going to be here for a little bit longer, which is fantastic. And thank you, Dr. Gabriela, for your input as well. So this has been a very fun talk, and I wish you all well, and I look forward to seeing you on the next talk. Thank you very much.
Thank you.
Thank you.